Hi guys, this is learning outcome number 4 of module 14. It's our last learning outcome of the last module. We made it, yay! So I'm going to try to make it as simple as possible. Here we're just going to be talking about the neurotransmitters, which we know already what the neurotransmitters are that are released in the autonomic nervous system, but more specifically the receptors that these neurotransmitters bind to that will tell you if the response is going to be excitatory or inhibitory. So let's get started. So based on the neurotransmitters that they produce and release, most autonomic neurons, they're going to be classified as either cholinergic or adrenergic. And the receptors for the neurotransmitters, they're going to be the ones that are going to be the integral membrane proteins that are going to be located in the plasma membrane of the postsynaptic neuron or the effector cell. So let's get started with differentiating the two different types of autonomic neurons. Let's start with the cholinergic neurons, which are the neurons that will release the neurotransmitter acetylcholine. So this is easy because acetylcholine has choline in it, and these are the cholinergic neurons. And in the autonomic nervous system, the cholinergic neurons are going to include all of the parasympathetic and sympathetic preganglionic neurons. Look at the images. All of the preganglionic neurons, they will release acetylcholine at the ganglion or at the adrenal gland. So they're all cholinergic neurons. In addition, we can see that most of the parasympathetic postganglionic neurons release acetylcholine. The sympathetic postganglionic neuron that innervates most sweat glands will also secrete acetylcholine. So they will be cholinergic neurons. And once acetylcholine is released from these cholinergic neurons, they will diffuse across the synaptic cleft, which is the space, and they will bind to specific cholinergic receptors, which are these integral membrane proteins located in the postsynaptic plasma membrane. Now, this acetylcholine, once it's released, it will bind to specific receptors. And we have two specific cholinergic receptors. We have what we call the nicotinic acetylcholine receptor and the muscarinic acetylcholine receptor. Let's start with the nicotinic one, which are going to be present in the plasma membranes of the dendrites and cell bodies of both the parasympathetic and sympathetic postganglionic neurons. It will also be present in the plasma membrane of the chromaffin cells of the adrenal medulla, because remember that the preganglionic neuron releases acetylcholine, so you need to have the receptor on the chromaffin cells so that they can bind to these receptors. And it will also be present in skeletal muscle, where at the neuromuscular junction, remember? So all these will have this nicotinic receptor. Now the nicotinic receptors, they're called nicotinic basically because the drug nicotine will mimic the action of acetylcholine by binding to these receptors. So that's why they receive the name nicotinic receptor. On the other hand, the muscarinic acetylcholine receptors, they're going to be present in the plasma membranes of the effectors because that's where they will synapse. So the smooth muscle of the organs, the cardiac muscle of the heart, and also in glands. And these are going to be innervated by the parasympathetic postganglionic axons. And in addition, we can see here that most sweat glands, they will receive their innervation from the cholinergic sympathetic postganglionic neurons, and they will possess the muscarinic acetylcholine receptor. And these receptors, they're called muscarinic mainly because there's a mushroom poison that's called muscarine that will mimic the actions of acetylcholine by binding to them. Now, it's important to bring up that nicotine does not activate muscarinic receptors, and muscarine does not activate nicotinic receptors, but acetylcholine does activate both types of cholinergic receptors. So this is really interesting, right? Now you would think, what is the effect of the binding of acetylcholine to these specific receptors? Well, the nicotinic receptors are going to be present in the dendrites and cell bodies of the neurons that are inside of the ganglion. So for the information to go from the preganglionic neuron to the postganglionic neuron and then to the organ, this information needs to be excited. It needs to be transmitted. So nicotinic receptors, they will usually cause or trigger this rapid neuro and neuromuscular transmission, because remember that nicotinic receptors are also present in skeletal muscle. So this is the response that you get when acetylcholine binds to nicotinic receptors. So it's going to be an excitatory response. On the other hand, the muscarinic receptors, they're mainly present in the parasympathetic nervous system. So remember that the parasympathetic nervous system is part of the rest and digest. So they're mainly going to decrease, for example, the heart rate and reduce the heart contraction force so that you can rest and digest. It is true that they're also present in glands, as we can see over here in the sympathetic nervous system, but in this case, they will also decrease the amount of secretion that occurs in the glands. And remember, when you get really nervous, sometimes you don't have a lot of salivation. Your mouth gets really dry. So this is the inhibition of the muscarinic receptor, or the effect of acetylcholine once it binds to the muscarinic receptors in the sweat glands. So it makes sense. Another thing that you can think about is how the parasympathetic is the rest and digest, and when you're eating and relaxed, your mouth produces more saliva. So it's the opposite effect of what occurs when you have the sympathetic subdivision of the autonomic nervous system acting on the salivary glands. Next, we have the other types of neurons, which are the adrenergic neurons. These will release norepinephrine, which can also be known as noradrenaline. So that's where the adrenergic part comes from. And most sympathetic postganglionic neurons are going to be adrenergic. So once norepinephrine is released from an adrenergic neuron, it will diffuse across the synaptic cleft, and it will bind to specific adrenergic receptors on the postsynaptic membrane, and this will cause either excitation or inhibition of the effector cell. Therefore, we can say that the adrenergic receptors, they're going to respond to the binding of either norepinephrine or epinephrine. In the autonomic nervous system, norepinephrine, it can be released as a neurotransmitter by the sympathetic postganglionic neurons, as we can see over here, or as a hormone by the chromaffin cells of the adrenal medulla, as we can see over here. And also, we have the release of epinephrine as a hormone as well. Now, the two main types of adrenergic receptors are the alpha receptors and the beta receptors. And in the autonomic nervous system, these receptors, they're going to be found on the visceral effectors, so on the organs that are going to be innervated by most sympathetic postganglionic axons, as we can see over here from this image, because only a few of the sympathetic neurons will innervate the sweat glands. Now, the alpha and beta receptors, they're going to be further classified into subtypes, which can be alpha 1 and alpha 2, or beta 1, beta 2, and beta 3. And this is going to be based on the specific responses that they will elicit and by their selective binding of drugs that activate or block them. So I'm sure that you probably heard, for example, of medications called beta blockers. Well, basically, they're going to cause the heart to beat more slowly because they will block the effect of epinephrine. So that's where the receptors come in, with this binding or selective binding of drugs that activate or block these neurotransmitters. Now, the alpha receptors, they will have a high affinity for norepinephrine compared to epinephrine, and beta 1 receptors, They basically have the same equal affinity for norepinephrine and epinephrine. Beta 2 receptors, they're going to have a high affinity for epinephrine compared to norepinephrine. And beta 3 receptors, it's going to be the opposite, where they're going to have a higher affinity for norepinephrine compared to epinephrine. The norepinephrine that's released in the sympathetic nervous system, it will function to increase arousal and alertness. It promotes vigilance, so it makes you more aware of what's going on. It enhances formation and retrieval of memory. It makes you focus your attention, and it can also increase your anxiety. So these are all excitatory responses. In the heart, norepinephrine will actually promote vasoconstriction, which is basically the narrowing of the blood vessels. And when you narrow the blood vessels, this increases your blood pressure. And therefore, this will also increase the heart rate. So this is your fight or flight, right? You need all this increase of heart rate. You need all this blood circulating through your body to be able to respond to an emergency. In addition, the epinephrine that's released as a hormone will also be one of the major hormones that's involved in the stimulation of the sympathetic nervous system for this fight or flight response. Therefore, it will cause similar responses as the norepinephrine. One difference between the somatic reflex, which was, for example, the arc reflex or the withdrawal reflex that we talked about when we mentioned the somatic nervous system, and the visceral reflex, which is the autonomic reflex, is in the efferent branch. So all this mechanism that we have been talking about. So the output of the somatic reflex is going to be at the lower motor neuron. So the one that's leaving the ventral horn of the spinal cord, and that will project directly onto the skeletal muscle and cause this muscle to contract. With regards to the output of the visceral reflex, this is going to be the two-step pathway that we have been talking about. So it starts in the preganglionic neuron that will emerge from the lateral horn of the neuron in the spinal cord, or the cranial nucleus neuron in the brainstem to a ganglion. And this will be followed by a postganglionic fiber that will project onto a target effector. So there are going to be several different types of visceral reflex, and here I want to talk about the pupillary reflex. So this is the reflex that we will focus on. First, we need to say that the activation of this pupillary reflex is going to come from the amount of light that's activating the retinal ganglion cells, which are these cells right over here, that will then come together to form the optic nerve. So if there's too much light, then your eye has a tendency of wanting to diminish that light that's passing through it. And this is what the pupillary reflex will do. So the output of the sympathetic system will project through the superior cervical ganglion. Remember, we talked about this? And you can see right over here that on the sympathetic nervous system, the first branch is going to the eye, whereas the parasympathetic system will originate out of the midbrain, and it will project through the oculomotor nerve, which is your cranial nerve number 3, all the way to the ciliary ganglion, which will then project to the iris. So the postganglionic fibers of either division will release the neurotransmitters onto the smooth muscle of the iris to cause the changes in this pupillary reflex. So norepinephrine will result in the dilation, and acetylcholine will result in the constriction. So this depends on which path it will take. So it's either the sympathetic pathway or the parasympathetic pathway or activation. So just as a review, this opening over here of the eye that allows the light to enter is the pupil. But what controls the size of the pupil is actually the iris, which will have a couple of muscles, and this will be the colored part of your eye. So the iris is made of two muscles. We have the radial muscle, which is a dilator, and we have the sphincter, which can also be called a circular muscle because it does go in circles as we can see from this image over here. And once it contracts, it will constrict and diminish the opening of the pupil. The dilators, which is the radial muscle, it will push this muscle out, and therefore it will dilate your eye, and it will increase the size of the pupil, as we can see from this image right over here compared to the original image. So basically what I want you to know is that if you have a dim light that's perceived, it will activate your sympathetic neuron at the level of the cervical vertebrae. That will then go to the ganglion that's located in the cervical region, so the cervical ganglion of the sympathetic chain. And then this will cause an activation of the sympathetic postsynaptic neuron to release norepinephrine, and the norepinephrine will then stimulate the radial muscles to contract, and when it contracts, it dilates the pupil, so it makes your pupil bigger, which makes sense because if you don't have enough light going in, you need to increase the size of the pupil to allow more light to enter into your eye so that you can see better. Now, the other pathway, if you have too much light coming in, then it will go to this specific nucleus over here that has this very strange name that's called the Edinger-Westphal nucleus. And this will then activate your parasympathetic neuron that will then go to the ciliary ganglion. Remember, the parasympathetic neuron over here goes to the ciliary ganglion, and this will activate the parasympathetic postsynaptic neuron to release acetylcholine, stimulating the sphincter muscle, which can also be called the circular muscle, and this will then contract and decrease the opening of the pupil. So you can see two examples of how the sympathetic and the parasympathetic autonomic nervous system will function to complement each other. One last thing that I want you to notice from this image is that the preganglionic neurons, they are myelinated. See how they have the myelin around them? And the postganglionic neurons are unmyelinated. This table is a good table for you to summarize everything that we have talked about with regards to the sympathetic and the parasympathetic divisions. So for example, with regards to the visceral motor neurons that are exiting the central nervous system, where are they located once they exit the central nervous system? Well, the sympathetic division will exit through the lateral gray horn between T1 and L2. On the parasympathetic division, there's a part that will leave the central nervous system from the brainstem, and there's another part that will leave through the spinal segments of S2 through S4. With regards to the location of the ganglia in the peripheral nervous system, we have that in the sympathetic nervous division, the ganglia, remember, will form this sympathetic chain ganglia, and it can also be located in these collateral ganglia, but what I really want you to focus on is that the sympathetic division has this chain of ganglia. With regards to the parasympathetic division, the ganglia is going to be located either on the target organs, remember, so they're actually located on the tissue of the organ, and these are what we call the intramural ganglia, or they're actually located very close to the target organ, so they're called terminal ganglia because that's where they terminate, close to the target organ. With regards to the preganglionic fibers, the length, we have that for the sympathetic division, the fibers are going to be very short and myelinated, and in the parasympathetic division, these preganglionic fibers, they're going to be very long because remember that the ganglion is located close to the organ. So this makes the preganglionic fibers have to be very long to be able to reach the organ or be close to the organ. And these will also be myelinated. With regards to the neurotransmitter that's released for both is acetylcholine on the preganglionic fibers. Now with regards to the postganglionic fibers, the length is the opposite. So because they have a short preganglionic fiber, the postganglionic fibers of the sympathetic divisions are going to be very long, but now they're going to be unmyelinated. It's the opposite for the parasympathetic division because they had a very long preganglionic axon. Now they're going to have a relatively short axon, and again, this will be unmyelinated. The neurotransmitter, usually we have norepinephrine, and for parasympathetic division, we always have acetylcholine being released at the postganglionic fibers. The neuroeffector junctions, here we're going to have these varicocytes that we talked about, which are these enlarged terminal knobs that will release the neurotransmitter near the target cells. But for the parasympathetic division, because these ganglions are sometimes located on the organs, there's going to be this neuroeffector junction that will release the transmitter to special receptors on the tissue surface. With regards to the degree of divergence, so when you have one neuron spreading out to several neurons, this is a divergence. For the sympathetic division, we have approximately one neuron affecting 32 different structures or different neurons, and the parasympathetic is one neuron for 6. So you can see how the sympathetic division actually branches out more, and therefore it can have an effect on more structures within the body compared to the parasympathetic division. In terms of general functions, we know that the sympathetic division is going to be your fight or flight. Therefore, it will increase your alertness, it will prepare you for emergency, and stimulate metabolism so you're ready for this response to fight or to flight. The parasympathetic division is the opposite. So it will promote relaxation, it will increase nutrient intake because you're probably digesting your food or relaxing or storing energy. And this is your rest and repose. There's also an acronym for this parasympathetic division, which is SLUD, where S is for salivary gland, L is for lacrimation, U is for urination, D is for defecation, and the other D is for digestion. So when you're relaxed, you have more saliva, you can actually cry, you can actually urinate, defecate, and then you digest. And I know that we're almost done with the semester, so right now I want you guys to activate your sympathetic division of the autonomic nervous system, and after we're done with finals, you guys can activate your parasympathetic division and just rest and relax and enjoy the benefits of working this hard.